Hi. Okay, so this video is going to be all about nucleic acids. And nucleic acids are one of the four major organic biomolecules that we are learning about. So we are going to title our notes Nucleic Acids. Sorry, hang on. Let me just straighten this out here. Here we go. Okay, and to go along with what we've been doing with all the other biomolecules, I want to introduce the monomers for this biomolecule so that you know what the pieces are that make it. So the monomers of all nucleic acids are called nucleotides. They're called nucleotides. And there are several different kinds of nucleic acids. I'm only going to talk about two of them today. And so the first nucleic acid that I want to talk about is DNA. And I know you have heard of DNA before. Do you know what the letters DNA stand for? They stand for deoxyribose nucleic acid. Okay, and so we're talking about nucleic acids. So that's what the nucleic acid stands for in the Na. And deoxyribose is a name of a sugar. So let me go into the nucleotide of DNA. And so the nucleotide are the monomer of DNA. DNA is a very large molecule and it contains information about all living organisms. So we all have our own unique DNA. Our own personal DNA are basically instructions on all the different proteins that you're going to make in your body. The proteins that you make you, you. Okay, the proteins that I make me, me. The proteins that I make me a human. And the proteins that this plant makes right here makes it a plant. And here's another plant over here, this orchid, and it has its own DNA and it is instructions for making all the proteins that make it an orchid. Okay, so let me go through what the monomer looks like, and then we'll talk about what DNA is more in general in a second. So the nucleotide of a DNA. Of a DNA molecule. Okay, so a molecule is a large polymer, and I'm just going to talk about the monomer. Okay, so nucleotides of a DNA molecule has three parts. So I'm going to list the parts and then we're going to draw it also. The first part of a nucleotide is called a phosphate group. Okay, and we've seen phosphate groups before. Do you remember where? We saw them in phospholipids. So in the video about lipids, there were phospholipids, and phospholipids have a phosphate group on one of the carbons in the glycerol. So nucleotides also have a phosphate group. Okay, and so I'm going to draw this. I'm going to keep it in the same color. So phosphate group, I'm going to do the same as I did with the phospholipid, which is going to be make a P and a circle around it. So that's the phosphate group. Okay, the second part of a nucleotide, I'm just going to change colors here, is the sugar. So a sugar, as you might know by now, is a carbohydrate. So carbohydrates are made of simple sugars, remember? And so the sugar in DNA is called deoxyribose. So for DNA, the sugar is called deoxyribose, and this is where the name comes from. Deoxyribose is the name of a sugar. And the way we're going to draw it is this phosphate group is attached to it. So we're going to put a carbon here. This is a carbon where the little bend is. And then we're going to make a bond to an oxygen. This is a bond to a carbon. This is a carbon right here. There's a carbon here, and there's a carbon here. Okay, so each of these corners represents a carbon. There is one carbon here, and these carbons in the deoxyribose sugar are numbered. I'm going to number them so that you can know this. This carbon here is number 1. This carbon here is number 2. This is number 3. This is number 4. And this carbon is number 5. okay? So there are 5 carbons in deoxyribose sugar, and that is the second part of the nucleotide in a DNA molecule, okay? So nucleotide is a monomer. So we have the phosphate group is one part, the deoxyribose sugar is the second part, and then the third part is called a nitrogenous base. Okay, nitrogenous base. And this is a molecule that has nitrogen in it, which is why it's called nitrogenous. And the nitrogenous base is always attached to the number 1 carbon of the deoxyribose sugar, so it's attached here. And there are 4 different nitrogenous bases for every nucleotide in DNA. Okay, there are 4 options. The 4 options are the following: cytosine, okay, and we always represent that with a C, or adenine, that's represented by an A. Thymine, that's represented with a T. And the 4th one is G, which is guanine. Okay, so any one of these 4 can be in a nucleotide. So I'm just going to make this an A, and I'm just going to write it as an A. So these are the 3 parts of a nucleotide. We've got the phosphate group, which is attached to the 5th carbon in the deoxyribose sugar, and then the 1st carbon is attached to the nitrogenous base. The base could be either an A, C, T, or G. Okay, so this is the monomer. The monomer is going to be attached to another monomer, attached to another monomer, attached to another monomer, and there are many, many of these monomers. And so what I want to point out is that a nucleotide, so I'm just going to draw this all in black here. So I have a phosphate group attached to a sugar. Okay, I'm just going to draw this kind of fast, and that's attached to an A. The next monomer, the next nucleotide is attached to this carbon here, which is carbon number 4. And so this is going to be attached to the phosphate group of the next nucleotide. So then I'm going to draw it like this. So here is the next nucleotide, and this could be a different nitrogenous base, let's say T, for example. And then this one's attached to the phosphate group of the next nucleotide. And this is attached to the sugar, and so on. So the nucleotides are the building blocks, and one nucleotide attaches to another nucleotide. Okay, so what I'd like to point out is that with DNA, as you probably are familiar with, it is commonly shown as like a twisty ladder. Okay, so I'm going to draw a twisty ladder like this. And so this is probably what you're familiar with when you think of DNA. And so DNA is this long, long molecule, and it has 2 sides. So it's called double-stranded. This molecule here is called a double helix. And that's because it has 2 strands, and it's in a twisty ladder, which is what a helix is. It means a twisty ladder. And it has 2 sides. So right here, what I've drawn is one side of the ladder, and what I'm going to show you is the other side of the ladder. Okay, so this, and this is what you need to know. You need to know that A always binds with T, and C always binds with G. Okay, those 2 always go together. So when you're making this ladder, what you need to know is that this A is always going to bind with T. So I'm going to draw the other side of the ladder on this side. A is always going to bind with T, and the other side is going to be upside down. Okay, so this side is going right side up, but this side is going to go upside down. So I'm going to draw this upside down. This is a little bit difficult to do, but I've been doing it for years, so it's no problem for me. But I just want you to get the idea that there's an opposite orientation. One's going this way, the other one's going that way. Okay, so we have an A goes with T. The T is going to bind to a sugar, so I'm going to draw the sugar upside down. So here's the oxygen, here's the phosphate group. Okay, and then T is going to bind with A, so I'm going to draw the upside down A. That's going to bind to a sugar. Here's the upside down sugar with the phosphate group. And this phosphate group binds to this part of that sugar. This C binds with G. Okay, so here's the upside down G, and that binds to the sugar, and that binds to the phosphate group, that binds to this, okay? So here is our ladder. And so what I want to point out with this twisty ladder here is one nucleotide is circled here, another nucleotide is circled here. Another nucleotide is circled here. Another nucleotide is circled here. So here I have 4 circled nucleotides, and here I've drawn 6 nucleotides. But what you need to know is that there is one side, and then there's the complement side. Okay, so if you know one side, you automatically know the other side. So for example, if I said that I had a DNA sequence of G, A, T, T, C, and I asked you, what is the complement side of this DNA? You would be able to tell me that G goes with C, A goes with T, A goes with T, T goes with A, T goes with A, C goes with G. So this would be the full double helix. Okay? Okay, so that is DNA. I want to then now introduce you to another nucleic acid, which is very important in biology, and that is called RNA. And RNA is very similar to DNA, okay? So what I'm going to do is I'm going to tell you, I'm going to explain what RNA is, and then I'm going to compare RNA to DNA, okay? So RNA is another nucleotide, and RNA stands for ribonucleic acid. Okay, ribonucleic acid. And RNA is also made of nucleotides, just like DNA is. And nucleotides of RNA are very, very similar to nucleotides of DNA. So a nucleotide of RNA contains the following. Nucleotide of RNA has 3 parts. The first part is a phosphate group. Okay, so I'm going to draw this just like I did with the DNA. So I have phosphate group, which is just a P with a circle around it. Okay, and so phosphate, as you can see, is very important. So it's very important that we have phosphate in our diet. If you remember from the chemistry video, there are 6 elements found in all living things. Phosphate is one of those things. Okay, that's because a lot of these molecules have phosphate in them. The second part of a nucleotide of RNA is a sugar. Okay, a sugar is a carbohydrate. The sugar for RNA is, guess what? It's called ribose. Okay, ribose looks almost identical to deoxyribose. The only difference is that in deoxyribose, there is one less oxygen, which is why it's called deoxy, because it's like eliminating an oxygen. So it's like ribose with one less oxygen. Okay, but otherwise it looks exactly the same. In fact, I'm going to pull in a picture so you can see it. Okay, so here is what they look like. I just drew them as just like a little pentose shape, but let me scoot over here. So let's see, this right here is deoxyribose in DNA, and I know that because it's missing an oxygen right here, where this one has an oxygen right here. So this is what's found in RNA. This is ribose. This is deoxyribose. And so you can see there's a carbon attached to another carbon, attached to another carbon, 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 and then there's a little oxygen at the top. Same exact molecule, except for here we've got oxygen right here. Let me redo that arrow right there. Okay? Okay, so we're back to our nucleotide of RNA. And so we have a different sugar for RNA. It's called ribose. And so I'm going to draw it like this. I have a carbon here, and then it attaches to a ring, a carbon ring. Okay, and these carbons are numbered. This one is also 1, this is 2, this is 3, this is 4, and this is 5. Okay, then the third part is again a nitrogenous base. Oops. Nitrogenous base. And the nitrogenous base is also attached to the number 1 carbon. And for RNA, there are again 4 choices. However, one of the choices is different than in DNA. Okay, so we still have cytosine, which is C. We still have guanine, which is G. We still have adenine, which is A. And then instead of thymine, we have a new one called uracil. And that's going to be shown as with a U. So I'm going to put a U here because uracils are only found in RNA, not in DNA. Okay, and so that is how you are going to be able to tell whether you're looking at DNA or RNA in a sequence, because if you see U's in there, then you know that it's RNA. If you see T's in the sequence, then you know it's DNA. Okay, so that is how you're going to be able to tell. Okay, and so the last point I want to make is with RNA, DNA had the double strand. So you have one strand attached to a second strand. RNA is not like that. RNA is single-stranded. So I'm going to write that here. I'm going to say, unlike DNA, RNA is single-stranded. Oops. Okay, and so instead of having the double ladder, it's just a one-sided ladder. So let me just kind of draw it for you. Okay, so it's just like this. It's just one side of a twisty ladder. And so instead of having the 2 rungs of the ladder, there's just one rung. It's just like this. It's like open. So RNA is used differently than DNA. DNA has information that has to be protected, and having the double-stranded keeps it protected. RNA is a temporary molecule that is just used to transmit information just temporarily. Okay, DNA is used to store information long term. RNA is used to store information short term. And so RNA only needs to be one-stranded because it falls apart quickly. So it does its job and then it falls apart. Where DNA needs to stay, protected because it's very important and it can't fall apart. You need your DNA. Okay, so the last thing I'm going to do is I'm going to show you a cartoon picture comparing DNA and RNA. Okay, so here is a picture from the textbook. And a couple of things I want to point out here is that, oh wait, that's too blurry. Hold on. On the left here is the DNA. And so like I said, DNA is a double-stranded double helix. So it's got 2 sides, where RNA has only single strand. Okay, so it's only one side of the ladder. Over here, all the way to the left is showing you the 4 nitrogenous bases for DNA, which is thymine, cytosine, adenine, guanine. These are all 4 different molecules. Thymine and cytosine only have 1 ring. Adenine and guanine have 2 rings. Okay, so over here on the right is RNA, and it has 4 nitrogenous bases also. It has uracil, cytosine, adenine, guanine. Uracil has only 1 ring, like thymine only has 1 ring. Cytosine has 1 ring. Adenine has 2 rings. Guanine has 2 rings. Okay, so that will come in and make more sense later when we talk about DNA. But that's all I have for today. Good luck on your quiz, and I will see you next time. Bye-bye.